here it's the first time I present in this huge room, and now I can hear myself. So I will be talking about the kind of work we're doing in Blue Waters by trying to use computer simulations to bridge different molecular timescales. So this is work that we've been doing at Stony Brook University. Ken Deal is the main PI. I'm a co-PI on this Blue Waters proposal. And so if we want to improve human health, we need to know more about proteins and the molecules that are working inside of our cells. So we all know about DNA. It has all the information about us. It's our genome. It has all the blueprints for all the proteins that are going to be acting inside of our cells. So DNA encodes for the proteins that we have. Those protein sequences, so proteins are these polymer chains, like kind of bits on a string, and each of those amino acids that compose a protein sequence, they have different physical properties. Once you try to satisfy all the physical properties inside those chains, the sequences will fold into a three-dimensional structure, such as this one, and those are the ones that are able to carry out some work. Now, what is important is that proteins inside our bodies, they interact with other molecules, and by interacting is how they perform their work. So they bind to other molecules, and sometimes they misbehave. So a big strategy is how do you design drugs that interact better with proteins in order to stop some kind of disease. So right now, it's pretty easy to get sequence information for DNA and for proteins, but it's very hard to obtain the 3D structures and the binding and detail, atomistic detail about the binding of different molecules. So there are experimental techniques that take very long time, they're expensive, and it's not always possible to treat all molecular systems. So a big way is, can we use computers to accelerate this process, and how do we go about it? So I just want to point out, like, the big challenge is right now there's about 100-time-fold difference between our sequence knowledge and our structural knowledge. So these are two different axes. We have about 100,000 protein structures that we know, and about 45 million sequences that are known. So what we would like to do is decrease this gap between our sequence knowledge and structural knowledge. And these numbers are a little bit outdated from 2014. So the way that we can treat these kind of problems in a computer is by looking at what's going on with proteins. So proteins are these long polymer chains, and they have, so the different amino acids have different properties. Here I'm showing you a pictorial view of the protein chain, and I'm showing 4 specific residues, amino acids that have special properties. So these are hydrophobic residues. They don't like to be exposed to solvents. So what their characteristics are going to do is they're going to try to clump up in the inside of a protein. And this is similar to what's going on inside our cells when a protein is synthesized. So it's made, and after some time, it will fold into a three-dimensional structure that tries to satisfy the physical properties of the amino acids that compose it. So what you are seeing is called protein folding, and what you are seeing is that those 4 residues that are hydrophobic, they don't like the solvent, have clumped up into the core of the protein. So we can put that in a computer. We can say what are the physical properties of different amino acids, how they interact, and so if we try to satisfy those interactions, we can make simulations that look something like that. Now the big problem is that we're trying to sample systems that have hundreds or thousands of atoms, and we are trying to simulate processes that are in the microsecond and beyond timescale. So these are very long compared to so we do numerical simulations, and the amount of time that we integrate is much smaller, so in the femtosecond timescale. So we have to simulate for very long. So if you just try a sequence like that to fold it on a computer, this is what a graph would look like. So we would like to fold this structure. We start very far away. It doesn't really matter what this graph is meaning right now, but like I don't know what happened there. Oops, sorry. So the higher you are, the worse structures that you are sampling. You want to go as low as this number here, 2 angstroms. So these are simulations that took about 6 months, and you see that you get nowhere near this 2 angstrom value. So we need strategies that are able to scale up better. And so this is the grand challenge that we face in protein folding in order to get at what the structures of proteins look like. So a different kind of computational challenge that you might be more familiar with is the game of chess and the game of Go. So in the 1990s, Deep Blue was able to beat Kasparov, the reigning champion in chess, by being able to calculate all the possible combinations of moves and selecting the best ones. So that was possible just because of brute force and a way of quantifying, given a move, what is the quality of the move. So what is the score? What's the score? Who's winning given a layout of the board? Now the game of Go, and we can quantify what is kind of the conformational volume, the amount of possible assignments in this game, which is 10 to the power of 50. Now the game of Go seems to be much more challenging. The board is much larger, and there's no clear way of, when you put a piece on the board, to know who's winning. So given a layout of the board, you don't know who's winning. You have to play out the games till the end. So supposedly this game, by brute force, you would not be able to solve it in a few years, but using some kind of advanced machine learning, AlphaGo was able to defeat the champions at this game. So if we make a parallelism with the protein folding world, in the 90s, late 90s, some people started, so Peter Coleman used the first microsecond long simulations to try to fold the Berlin headpiece, a very small protein, and they got pretty close to it, but not really to the native state. And this is a small kind of protein. If we simplified what the conformational volume would look like, it's 10 to the power of 30, so even smaller than here. And after 20 years or so, what we're able to do is slightly better. We're getting at proteins that are now double in size, but still we're not able to get to the large proteins that we would like to get to. So things that are 300 residues or whatnot, like proteins that are, so the average size of a protein in a human is about 300 residues, and we are nowhere to folding things that are of that size. So the big problem is the scaling problem. The more particles you have in the system, the more atoms you have, the worse you scale up. So physics has solved the problem of 2 particles interacting. Beyond that, you just have to do numerical integration. So this is how we scale when we use a brute force approach. So this is us right now. We're able to fold proteins that are around 30 to 40 residues, and we would like to go all the way up to 300 residues. And it's not going to happen anytime soon. So what we have been using in Blue Waters is a technique we call MELD, modeling employing limited data, which scales much better, but still not as fast as we would like. So we're solving some proteins that we could not solve before, and we're trying to improve the methodology to go even farther. So how does this methodology work? So under the hood is molecular dynamics, so it's using Newtonian physics to integrate the equations of motions through time. And the advantage in MELD over traditional molecular dynamics is that we introduce data. And in particular, we use data that is sparse, ambiguous, and noisy. So when you have a lot of data from experiments, you don't need to do simulations. In the limit where the data is not good enough on its own to solve the structures, you need some kind of integrative approach that uses the data and the simulations. So what do I mean with data that it's ambiguous, noisy, and sparse? Imagine that you go to the beach, and then as you enjoy the day, you go back home, go to your car, and you say, oh no, I lost my keys. Now you have to find them in the beach, and there's no way you can go through all the beach and find them. So the first thing you'll do is, okay, I was laying in my towel right over there, I went to the bar, and then I was walking along this path. So now you know 3 places where your keys are likely to be, right? So the keys cannot be in all 3 places at the same time, so the data is noisy. So 1 out of 3 places is going to be correct. At the same time, it's ambiguous because, you know, even if you were sitting here, like there's some uncertainty about how close to this area the keys are going to be. So there's going to be some spread. So the way, but even so, this kind of information is going to allow you to find the keys much better than if you didn't have it. So we use the same for molecular simulations. And we use a Bayesian inference approach to integrate the data into the simulations. So in a Bayes theorem, and I will go really fast through this, if you have details, there are a couple of posters here that you can ask questions to. I will tell you about the posters later. So you have a prior that is your force field, your physical simulations. They have a landscape that is very rugged, they're very hard to explore. And you have the likelihood. It's how well the structures that you're sampling agree with the data that you have. So since it's noisy data, you only try to satisfy subsets of the data. And what you're creating is a much more funneling surface. So when you combine those two things together, you're efficiently sampling from this kind of landscape. So it's very funneled energy landscape. And what you want to see is what is the probability that I'm in one of those big minima. Now, since this is very funneled, you're going to have problems exploring this landscape. So the way that we get around this is by using a replica exchange protocol. So what that means is that instead of having a single copy of the system and just simulating for a long time, now we have multiple copies of the system. So in Blue Waters, we have the same simulation multiple times, and in each of those simulations, we run slightly different conditions. And every now and then, we allow the conditions to swap under certain probabilities. So efficiently, what we're doing is, in some of the replicas, we're running at high temperature, and we are not imposing the data, whereas some other, we are imposing the data very strongly, and we're using low temperature. So efficiently, effectively what we're doing is, at high temperature, we're sampling from this very rugged energy landscape, and we are going at high temperature, so it's very easy to go all over the landscape. As we anneal down, we're going to be funneling more and more so that you get trapped into these different minima. And the nice thing is that we can use statistical mechanics to get back what are the relevant states. So yes, looking at populations that we sample, we're able to know which of these minima is the most important. So we have used that several times to predict the structures of proteins. One of the most interesting things we do that we can only do in Blue Waters is participate in a blind contest or competition called CASP. So the idea is that you are given a protein sequence and some time, and you have to predict the 3D structures. So here I'm comparing our blind predictions with the experimental structure. And even if you have never seen a protein before, you can see that the overlay is quite good. And these numbers that I'm showing you here is what's the resolution that we're getting. So we're getting resolution at the Armstrong level, so 10 to the power of minus 10 meters. And so how do we compare with other methodologies? So physics-based approaches have not been able to solve this problem before, so we can only compare with bioinformatics methods. And this competition provides the best scenario for that. There are about 200 different groups participating in CASP. Each one of these lines is a different CASP group prediction. So even though these plots are a little bit weird, what you want to have is the shoulder of this curve to be as far to the right and as low as possible. That tells you that you have a big percentage of the structure that is very accurate. And so what I want you to see is that for this protein, even though we did really good, there were some bioinformatics methods that did better. In this one, we were amongst the top, but still, a few were better. But for this one, we're better than any other kind of bioinformatic method. Bioinformatics, the main thing that these methods do is they look at similarities in things that are known and sequences that we need to predict. And then they copy the structural features. So they cannot escape our current knowledge. Whereas these kind of physics-based methods can. So in this way, Blue Waters has been key for this competition because it runs on for about 3 months, and we have new targets every day. There are about 200 different competing groups. We have hundreds of proteins that we have to simulate, and we have very tight deadlines. So for every sequence, we have at most 3 weeks to give back our solution, but sometimes we're given as little as 5 or 7 days to predict the structure. So we need a fast turnaround time. If we have to wait in the queue for 3-4 days, we're not going to be able to predict anything. And so the nice thing is that we are currently the only physics-based approach that participates in CASP with these very tight deadlines. And so what the nice things that we can do is that we use about 30 GPUs per protein that we simulate. We have sparse communication between the nodes, and so we have had a lot of help in how to optimize these to run efficiently and work in the queues in blue waters. And one of the nice things is that this software was highlighted as being highly effective in GPUs, using about 90% of the GPU cards. And so this is just one aspect of the things that we do in blue waters with our allocation. The other things that we do is trying to go to the next step. How do we see how molecules interact with proteins? So what I'm going to show you here is a simulation of different molecules called peptides interacting with a protein named MDM2. So this is a protein involved in cancer. And what we are trying to see is compete different molecules and see which one of them like to bind better with this protein. So what you are going to see is that the two molecules go near and far from the protein. Every now and then, they identify a place where they like to interact. They stay there for a while. They're kicked off after a while. And just by running these long simulations, you can just get a sense of, using statistical mechanics, what are the best peptides, what is the affinity they have for that interaction. And that helps in designing better drugs. The other thing that we do is we look at pathways. So we made simulations. We know what are the relevant states. Now, do we know how to get from one state to another? So what I'm showing you here is a very small example, a toy example of a helix-to-helix transition. You have a left-handed helix that converts into a right-handed helix. And what I'm showing are all the possible states in the system and the transitions between them, which we can calculate in an embarrassingly parallel way in blue water. So basically, we use our MELD approach to calculate states, and then we use short individual simulations that start from each state to get transition counts. And the nice thing about this project is once you have all the transitions converge, you can start looking at what are the main flux pathways. So this tells you, okay, right now, if you want to go from one state to the other, there are two main pathways. You can either go to this lower route or this upper route. And the meaning of that is, if you have a helix, you're going to open it up through one end and go all the way up, or you're going to open up from the other end and go all the way down. So that's what this plot is showing you. And I went a little bit fast through all of this, but there are a couple of posters that are going to be later on today. So while you enjoy your drinks, you can also look at the poster by James and the poster by Emiliano. They will be talking about this methodology in trying to get at proteins that cannot be captured by informatics and at different approaches of protein-protein interactions and whatnot. So with that, I just want to thank Ken Dill, who is the PI on this grant, Justin McCallum, who is a co-developer of the code, the Dill Group, Blue Waters, the Loeffler Center, and I also want to advertise that I'll be starting a position at the University of Florida. So if anybody is interested in this kind of work, there are postdoc positions there. So let me know. Thank you.